A single strand of DNA is practically a ghost. It holds so little physical mass that trying to observe or study it on its own is mathematically impossible. But finding that genetic ghost is often a clinical necessity. If a doctor needs to know whether an active virus is hiding in a patient, they cannot simply place a nasal swab under a microscope. There is not enough genetic material present to trigger a readable signal. Now, the challenge is one of scale. How do you take a microscopic target you cannot even see and force it to copy itself billions of times until it becomes loud enough to measure? Without a mechanical process to rapidly mass-produce that invisible genetic code, our ability to sequence genomes or diagnose viral outbreaks would essentially be blind. The solution to this duplication problem is a specialized biochemical toolkit known as the master mix. This chemical setup functions like a coordinated industrial operation. As the team in our source footage puts it, "All right, team, we need the master mix balanced." This diagram isolates 4 specific components. We start with DNTPs, the nucleotide building blocks of DNA. Next, magnesium chloride is added as a stabilizer. Then come primers, targeted scouts finding the specific gene. Finally, the heavy machinery, Taq polymerase. But there is a mechanical paradox at the center of this process. To build a perfectly new genetic sequence using these ingredients, the biological machinery must first break the existing one apart. That separation begins with phase 1 of the thermal cycle, denaturation. We trigger this by rapidly spiking the temperature of the mix to 95 degrees Celsius. At that extreme heat, the weak hydrogen bonds holding the 2 interior sides of the double helix together are forced apart, while the strong outer structural backbone survives the heat. Marking the moment the double helix surrenders its secrets, the DNA officially splits into 2 fully exposed single strands. Immediately, we drop the temperature to 55 degrees Celsius, initiating phase 2, annealing. Those small primer blocks rush in, locking on to precise designated locations. That cooling threshold is mathematically calculated. It is called the melting temperature, and establishing it ensures that the primers only bind to their exact targets, preventing them from snapping on to incorrect genetic sequences. By shifting rapidly from a near boil to a cool state, we transform a chaotic genome into precisely marked construction sites. The environment warms back up to 72 degrees Celsius for the third and final phase, extension. 72 degrees is the exact optimal operating temperature for the Taq polymerase enzyme. While most proteins are permanently destroyed by high heat, Taq was originally sourced from extremophile bacteria living in boiling hot springs, allowing it to survive the initial 95-degree spike. Functioning perfectly in the heat, the Taq enzyme binds to the primer construction sites and begins pulling in the raw nucleotides, assembling a brand new complementary strand at a rate of 1,000 bases per minute. As the factory team notes, Taq polymerase builds a new world. At this exact moment, 1 complete DNA molecule has successfully become 2. These 3 simple temperature shifts, heating, cooling, and warming, operate as a microscopic biological printing press. The true utility of this process lies in its recursion. The moment those 2 new strands are completed, they immediately serve as the templates for the very next thermal cycle. This chart tracks the resulting mathematical loop. The growth starts slowly as 2 strands become 4, and 4 becomes 16. But because every copy generated is itself a new template, the curve quickly becomes vertical. After exactly 30 cycles, taking only a few hours inside a modern thermal block, that single invisible genetic sequence has been perfectly replicated over 1 billion times. The chemistry makes the single copy possible, but it is the deployment of exponential mathematics that allows us to overwhelm the microscopic scale. When biochemist Kary Mullis conceptualized this reaction loop in 1983, it provided the mechanism to bypass the primary bottleneck of early biological research. Today, when a scientist finishes running those 30 cycles, they must still verify that the billion-fold multiplication actually occurred. They verify this by loading the final mixture into an electrophoresis gel. If the amplification was successful, the accumulation of duplicated DNA creates a physically dense, bright pink band that glows under UV light. Mission accomplished. 1. That physical glowing stripe is the confirmed delivery of the targeted sequences. PCR forces the microscopic world to scale up into the visible, acting as the fundamental bridge connecting the code of life to human comprehension.